Tonight, we're delighted to welcome Patrick Holford to the B12 deficiency support group. Regular Monday meetings. These meetings happen every Monday. Website is www.b12d.org. And the way you find out about these meetings is to join our mailing list, which is to join as a member, which also allows you to leave your readings, your vitamin B12 deficiency symptoms over a number of times. Then you can look back on your history and see how your symptoms have developed over time. We're delighted to welcome Patrick Holford from Food for the Brain. His topic is B12 deficiency driving cognitive decline and mental illness? Vitamin B12 is unique in that it's only found in foods of animal origin and depends on stomach secretions to be absorbed. Patrick Holford, founder of the Food for the Brain Foundation, foodforthebrain.org, presents new evidence that B12 deficiency is far more widespread than realized and is driving all sorts of mental and neurological problems, including cognitive decline and ultimately Alzheimer's. This webinar is essential for any healthcare practitioner interested in mental health. And we always welcome people who are either suffering from B12 deficiency themselves or caring for someone who's suffering in order to understand better themselves how to manage it and to find some support from other people. I'm going to hand over to Patrick now. Thank you.
All right. So yes, thank you to Hugo and your team at B12d. This is such an important topic. I am a psychologist originally, and some people think of me as a nutritionist, but only because my interest is in the brain. And very specifically, I'm interested in that space between the ears. Really, this is the final frontier. We understand how our muscles work and our heart work, but most of us are not really aware of exactly what's going on between the ears and how that functions. It's just another organ, and there are straightforward dynamics. And of course, the brain is remarkably dependent upon nutrients, one of which is B12. I am lucky to head the Food for the Brain Foundation. We have an extraordinary world-class team of experts working and supporting the Food for the Brain Foundation, from Professor David Smith, whose research on B12 and homocysteine is really extraordinary. Professor Jin Tai-Yu is really the top man in China. Professor Peter Garrard, arguably Britain's leading neurologist at St. George's, University of London Neuroscience Research Section. We have so many others. Julia Ruckledge is a professor who specializes in children's mental health. Some of you will have heard of Robert Lustig. He's our sugar man. And, you know, the list goes on and on. Heading our research team now is Dr. Tommy Wood, assistant professor of pediatrics and neuroscience at the University of Washington. So we really have some top people in our team. I want to start a brief film. Everything I'm going to show you relates to B12 and gives you some piece of information that will be important to unraveling how this nutrient has such a big effect.
Brain has 100 billion brain cells called neurons, making 100 trillion connections between them, communicating to each other with chemicals called neurotransmitters. Brain cell membranes are made from both phospholipids, which are abundant in eggs, which attach to the omega-3 fat DHA, abundant in oily fish. To make a brain cell membrane, they have to be bound together by a process called methylation. Methylation depends on B vitamins. If you lack B12, rich in animal foods, or B6 or folate found in whole foods and greens, your blood homocysteine level rises. Your homocysteine level predicts the rate of brain shrinkage as you age. Lowering homocysteine with B vitamins cuts shrinkage by over 70% in those with enough omega-3.
So immediately we're learning about the importance of phospholipids, omega-3, and methylation dependent on B vitamins, including B2. And the sad tragedy is that if a person is diagnosed with dementia and has a raised homocysteine level, unless they're diagnosed with pernicious anemia, they are not allowed to be prescribed. A doctor cannot prescribe B12 to someone unless they're actually diagnosed with pernicious anemia. It's extraordinary. So let's take a look at this whole area of cognitive decline and dementia, which is a major, major issue. A recent report in China says if they do nothing by 2035, the cost to China will be $1.8 trillion, which is half our GDP. So in the UK, we have 790 people. That is 7 double-deckers buses worth of people diagnosed every single day. And globally, 3 seconds, every 3 seconds, someone is diagnosed with this preventable disease, as I will show you. And what's also becoming clear is the beginning of this cognitive decline. And by the way, Alzheimer's is indicated by a shrinkage of a central part of the brain, which is actually the hippocampus. You already see shrinkage in teenagers who have metabolic syndrome. In other words, they've been eating too much sugar and carbohydrates. And you see a loss of cognitive function, not only in the teenagers, but also in middle age, in adults age 40, for example, who have a normal blood glucose, but within the top end of the reference range. You're already seeing a 14.5% increased risk for Alzheimer's disease. So this isn't something that suddenly happens when you hit 70 or 80. It's creeping up on you. Now, I'm going to move quite fast, and I'm going to give you lots of references. And I'll also send the PDF of this presentation so you can see the slides without having to write everything down. But most of the references are very nicely organized at foodforthebrain.org/PHC references. PHC is the Public Health Collaboration, which is a group of switched on GPs who realize that eating too much sugar is a major driver of disease. So .org/PHC references, you'll see the references with links to the studies organized so you don't have to be scribbling like crazy. When a person starts to lose their cognitive function, the first sort of level of it is called mild cognitive impairment, or MCI, or pre-dementia. And if it's worse than that, it is called dementia. That's how you diagnose dementia, on the basis of a cognitive function test. And we at the charity got permission to digitize this very cognitive function test that is used worldwide in memory clinics. And we did a study validating it. That is the digital version versus the pen and paper version used in memory clinics. And it is exactly the same. So this is a validated, accurate cognitive function test that actually can pick up changes 40 years before a diagnosis. And we have tested, I just looked, we just hit 410,000 people have taken the test. Now, that doesn't diagnose you with Alzheimer's. Alzheimer's is a disease where a central part of the brain called the medial temporal lobe, which is where the hippocampus is, shrinks. And that test, that scan, which was developed originally by the team at Oxford University, headed by Professor David Smith, that scan is necessary to diagnose Alzheimer's dementia. And I'll talk a bit more about that. What we're looking at here is 2 people who've had MRI scans, and then a second scan after 6 months, which is superimposed over the first. On the left, there is no change. On the right, what you're seeing in black, in the central part of the brain, the medial temporal lobe, is the brain cells that have been gone. You know, they've died. They have died in a 6-month period. That is what Alzheimer's is. The person on the left has a homocysteine of 7.8, which is quite normal. And the person on the right has a homocysteine level of 13.1. This is a blood test. It's something you can measure. And for sure, if your level is above 11.5, we can say there is accelerated brain shrinkage. But really, you do not want to be above a homocysteine of 10. And I'm going to talk about how that relates to B12, what makes it go up and what makes it go down. So really, this process that we're seeing in, well, we would call it normal aging, that the whole brain, once you're in your 70s or 80s, a bit like height, reduces by about 0.5% per year. Perhaps that's not even necessary. There are so few studies on people who are in optimal health. We have to find out. In mild cognitive impairment, you've got a brain shrinkage of about 1%. And in Alzheimer's disease, the total brain shrinkage is about 2.5% a year. So we're talking about an increasingly shrinking brain. And this degeneration, this shrinking in the medial temporal lobe happens some 40 years before a diagnosis. But of course, it's not scanned for early. So by the time people have scans, it's already, for many, simply too late. So the first message is we want to be testing people, from age 40, really, to be able to pick up subtle early changes, to be able to do something about it. Now, here's something, really quite extraordinary. If you look at humans split from, our ancestors split from bonobo and gorilla and chimpanzee about 6.5 million years ago, they maintained and have a brain weight of around 350 cc's. And gradually, through evolution, our brain size increased through up to the Neanderthals and then into Homo sapiens about 29,000 years ago. And around 29,000 years ago, we start to see brain size of 1,660 cc's. So, substantially higher than that of chimpanzee. And what's quite extraordinary, and by the way, this is done on cranial size, so it's ancient skulls basically. Today, the average brain size is 1,336 cc's. So we have in the last 29,000 years lost something like 20% of our brain size. And really what makes us human is not our brawn, it's not size, muscle, strength, it is the brain. And in a newborn baby, there's an extraordinary high brain to body ratio. So babies have to be born with big brains. It's all about the brains. That's what gave Homo sapiens their edge. There is a brilliant book just out called The Shrinking Brain by Professor Michael Crawford and David Marsh. And anyone seriously interested in mental health needs to read this book. The message is very clear because what's totally obvious, if you look at all of the evidence, is that we are the waterside ape. We are that ape that learns to exploit the wetlands, the swamplands, the rivers, and the coast. I mean, they're full of food, really easy food. You don't need to go hunting animals when you can just pick up oysters and mussels and clams and crabs and so on. I'm sure, as David Attenborough once said, that we may have been watching the storks and the birds eating all this lovely, rich seafood, and we started to do the same. And in the process, we became upright, wading in water. We developed manual dexterity, opening shells. We developed a layer of insulating fat, which for many of us has become rather large, a hairless body, a nose for swimming. Infants can swim a long time before they can learn to walk. ENT specialists know that there are these little protrusions in the ear called extoses, misspelt in this slide, extoses, that protrude. And if they find these protruded in the ear of a patient, then they say, you're a diver, aren't you? Because if someone spends a lot of time diving, these extoses protrude. And in the case of the dolphin, which has every single bone that we have in our hands and arms in its flippers, its extoses have completely blocked the ear, which is fantastic for diving, lousy for hearing. So they've developed sonar as an alternative system. But the real clincher came when David Attenborough said, when a baby is born, they have this waxy waterproof layer, the vernix. I have never seen this in any land-based mammal. And he asked on television, I wonder if it occurs in the sea mammals. And a French-Canadian marine biologist heard this and investigated. And sure enough, we have identical vernix to that of a seal or a sea lion or those other marine mammals that evolved on the land and then decided to move into the sea. And this diet, this marine food diet, not only that, because we would have been eating plant foods, land foods, and water foods, is very rich in B12 and omega-3 and iodine and selenium, all sorts of very important nutrients, which I'm absolutely convinced were completely essential for the evolution of the human brain. We didn't quite make it back into the water, so to speak, but there are tribes in the Indonesian Ocean, the Moken and the Baju, who live on villages floating in the sea. They swim for 5 hours a day. They're born in the sea. They're weaned on seaweed, and they can hold breath for almost as long as a dolphin. They're actually adapted in this direction. So we didn't quite make it. Where I live in Wales, they discovered the oldest complete Homo sapiens skeleton, 40,000 years old, and it was covered in jewelry made of shells. And therefore, they thought it was a woman, but actually it's a man. And the story, because this is just sort of the way it's told, I remember hearing about this on the BBC, is they would have been wandering the plains, catching antelope with spears, etc., etc. But why would they bother? I mean, the population was so small in those days, and all of the estuaries and coastal waters were so rich in nutrients. Anyway, on analysis, they found that 20% of their diet was seafood. And if you think that they were expending 2 to 3 times the calories every day that we do, hunting and gathering and chopping wood and walking and so on, it would really mean that half of our entire diet would have to be seafood to get to the same level of B12, level of omega-3, level of phospholipids that our ancestors have probably eaten during the entire time of our brain's evolution up until we started to switch to land-based agriculture about 10,000 years ago. That's really the turning point. Now, what happened was Professor Michael Crawford analyzed the types of fats in about, I think it was 36 or 38 different species of animals, and they vary depending on what they eat and where they live and so on, except for the brain. The brain of every single animal, without exception, was remarkably rich in 2 fats. The first is a type of omega-3 fat called docosahexaenoic acid, DHA, and the second is an omega-6 fat called arachidonic acid. All brains of all animals principally use those fats as structural fats that are essential for brain function. If an animal has a lesser supply, they have a smaller brain, but the brain contains those fats. And we start to learn that this omega-3 is terribly important for mood. And in medicine, we want evidence-based medicine, which means lots of randomized placebo-controlled trials pulled together into a meta-analysis. We already have that. And it's very clear that omega-3 fat is exceedingly effective for depression. Professor Joe Hibbelin, who's one of the world's experts on omega-3, he used to be in charge of the mental health of the US Navy. He gives his depressed patients, as a psychiatrist, 4 grams of omega-3 and says which is very consistent with what we may have eaten, that they feel fantastic. They feel very content. And he said they also have a much better sex life. They report much better orgasms. I'm not just saying that flippantly, because there had to be a reason why these land-based mammals decided to permanently commit to the sea. And it will have been to do with their brain and their mental health. And they think they just felt better. And, here we are going in the opposite direction, where currently in the UK, in the poorer areas of the UK, 50% of women are now on antidepressants. So we've really gone from Homo equaticus to Homo sapiens to Homo fatians to Homo caffeans to Homo apians. And there is no question that we are, as a species, devolving. We are losing those very things that make us human, our intelligence, our connectedness, our empathy, and so on. So when you look at the whole pie of dementia, 62% is called Alzheimer's, 17% is vascular dementia, which has exactly the same drivers and prevention factors as Alzheimer's, as I'll show you. And 10% is mixed, which usually means vascular plus Alzheimer's. So we're really looking at 89% of dementia is driven by the same things, which I'm going to show you, and can be prevented. Now, there is, of course, this big idea that genes cause disease. And to be honest, if you go right back to Darwin was a very bright guy. And he said there are 2 drivers of evolution, and one is the conditions of existence, in other words, the environment that species evolve in, and the second was natural selection, survival of the fittest, so to speak. And of those 2, he said the conditions of existence were the more important. However, despite the fact that he said that in every one of the 6 editions of Origin of Species, it's sort of been written out of the history books, because the imperialists, they love the idea that white was better than black, or that the Aryan race was superior, and somehow we had that edge. And then, of course, when DNA was discovered, everyone got extremely, excited and thought everything is driven by genes. But it's just not true. It's been one of the really big mistakes we're making right now, is all this emphasis on genetics and so little emphasis on environment, that is the conditions of existence, that is nutrition. So the reality is there are 3 causative genes, AP and the presenilin genes, and they account for less than 1% of cases of Alzheimer's. So less than 1 in 100 cases of Alzheimer's is caused by genes. And if you're unfortunate to have those genes, you will develop problems early, in your 50s or 60s. Now, there are other genes that may predispose, but they don't cause. And genes are just instructions to make proteins, usually enzymes. So if you have the ApoE gene, which we all have, but you happen to have the ApoE4 version, your chemistry is a bit skewed in some places, and you will have a 4 to 6% increased risk, so to speak. But the point is that all genes which are making enzymes, for example, only exert their effects through biological mechanisms. And those biological mechanisms depend on nutrients like B12, omegas, et cetera, et cetera. So what happens is that these don't cause disease. They just sort of skew you in that direction, but they are like a dimmer switch. You can turn that genetic effect down. And we know that eating a low glycemic load diet, having more B vitamins, especially B12, vitamin D2, K2, omega-3, quercetin, resveratrol, all of these actually dampen down the effect of ApoE4 quite considerably. Now, in all the big studies on dementia, cognitive decline, and so on, of course, the authors will have measured ApoE status. About 20% of people have the ApoE4 gene. And a nice example of this early on in this year was at the British Medical Journal. This is the study of GIA. And by the way, I want to tell you right now that the biggest set of data relevant to dementia is the UK Biobank. But I think sometime next year, especially with your support, our database will be bigger than that. And it's the Chinese who are paying the UK Biobank for their data. And this study is a classic example of 29,000 people followed over a decade. And those who had a healthy diet and lifestyle were 9 times less likely to develop dementia. Another very good study called the FINGER trial, which much like what we're doing at Food for the Brain, there's nutrition advice, advice to exercise, to use your mind, social interactivity, et cetera, et cetera. And, a very effective intervention, so to speak. But when I look at these studies and I look at ApoE4 and I ask the question, does having an ApoE4 status of this gene make any difference to the outcome? The answer invariably is no. I've actually only found one study where it made a difference to the outcome. So I think of these predisposing genes like a weak light. And in the darkness, it has a small effect. But in the light, when you change diet, you improve your lifestyle, you take B vitamins or omegas or whatever, you can't see the effect anymore. So, here we are in a situation of a disease that is largely preventable. And the reason I show you that figure is a recent sort of piece of pure statistics on the UK Biobank data, looking at behaviors and circumstances. And Professor David Smith and Professor Jintai Yu, 2 of the members of our science board, our authors in this paper, estimate that up to 73% of dementia cases can be prevented. I think it's more because the nature of this study is they weren't looking at blood levels of anything. They were just looking at behaviors, whether you smoked, your level of education, things that were recorded by the UK Biobank, which did not record, for example, homocysteine. But generally, we are told, if you read the media, that it's 40% is preventable. And this comes out of a ongoing document called the Lancet Commission, authored by Professor Jill Livingston. And the thing about this report, which started in 2017 and was updated in 2020, is, despite being sent all the evidence on omega-3 and homocysteine and B vitamins, the author has chosen to simply ignore it. So we have the second in charge at Oxford Medical School providing unequivocal RCT data, randomized controlled trials, again and again and again, and they are simply ignored. And, you know, there lies the problem. We haven't looked at the big targets, the ones that make the biggest difference. In fact, the best meta-analysis to date of 243 observational studies, 153 randomized controlled trials, is by Professor Jintai Yu. And in it, quite rightly, from the statistics, he says homocysteine-lowering treatment with B vitamins, and B12 is the key, seems the most promising intervention for Alzheimer's disease. And he shows this in a very, very nice way, a sort of curve of all the things that would happen to gradually lead you to the point where you are very, very likely to get Alzheimer's. And the dotted line in the middle, if you're very high above that, it increases risk. So you can see depression is high up. And if it's a yellow dot, it means the level of evidence is level A, is strong. So hyperhomocysteinemia, high homocysteine, depression, diabetes, these are big risk factors. And down below the dotted line, you can see cognitive activity, use it or lose it, high level of evidence, very protective. So it's a nice way to show this kind of data. If you go to the US National Institute of Health, they've done a similar thing looking at the attributable risk for Alzheimer's, and they attribute 22% of the risk of Alzheimer's to a raised homocysteine level, a lack of B vitamins, 22% to a lack of seafood, and a lack of omega-3. Now, you can't just add those two together because some people will cross over. Some will both, you know, the numbers don't add up to 100% because you could both smoke and eat fish and, you know, have low B12 levels and so on. So really, in these sort of apparently complex diseases, we have to look in a different way, not the usual reductionist approach, breaking everything down into little pieces, but a systems-based approach to understand that we have to look at the structure of the brain, the function of it, and the utilization. You know, this is, it's really like, you know, we have parts, the parts relate, and then there is this life flowing through a living system, which is the utilization. And only when you really start to think in that way does it become obvious why things like cataracts or hearing loss or obesity or you know, ketogenic diets, which are beneficial, or diabetes or head traumas. You can understand why each of these is a piece of a jigsaw, which in combination can lead to this problem. And that is also why there never will be a drug. There's absolutely no way there is going to be a drug that will cure this condition, you know, or ultimately prevent it, because there is no one place where this is happening. And our group of experts, you know, collectively, we made the decision to organize the risk factors into 8 domains. And I'm going to be particularly talking about the B vitamin domain, but also the brain fats, because as we saw in the film, one cannot work without the other. And I'm going to show you exactly why that is. So first of all, let's get deep into homocysteine. And I made a little film to explain how it works, but I'll break it down.
Food protein contains an amino acid called methionine, which turns into a toxic amino acid called homocysteine, en route to making SAMe, which is the good guy, a methyl donor. The dots here are the methyl groups. They help make neurotransmitters and phospholipids, control genetic expression, even repair DNA. Where do the dots come from? The answer is food, folate. It's a B vitamin rich in greens. You also need B12 and B3 and B2 and B6. And there's another pathway that needs TMG and zinc. If you don't have these nutrients, you can't do methylation and your homocysteine level goes up.
Yeah, so what's happening really is we're eating an amino acid, methionine, and we turn it into homocysteine, which we don't want, en route to making S-adenosyl methionine SAMe. And SAMe does something called methylation at a rate of about 1 billion methyl reactions every 2 seconds. So really, the nutrients we get in food largely don't work until they're acted on in some way. And methylation is the critical way in which we turn one thing into another. Like if the fire alarm goes off in 0.2 of a second, you're pumping adrenaline. How do you make adrenaline? by methylation. How do you detoxify so many things? Like histamine, if you have too much, you detoxify by methylation. Methylation is how you turn genes on or off. It's how you repair DNA. There isn't a specific need, so to speak, just for folate in pregnancy. You know, all women are recommended to have folate. It's not the folate, it's the methylation. And what happens is that if you are lacking in B12, you can't convert the toxic homocysteine into SAMe. And if you're lacking in folate, you also can't do it. So you need B12 and you need folate. And if you don't have them, your homocysteine goes up. And you not only need that folate and B12, but also that SAMe, the methyl donor, is going to attach the phospholipid that the film said is rich in eggs, also rich in fish, to DHA. So your brain is actually made out of what we call phosphorylated DHA. Phospholipids attached to DHA, done by methylation, dependent on B12 and folate. There is actually another pathway whereby homocysteine can be routed to the left and to produce glutathione, which is the body's most potent antioxidant. And that pathway is dependent upon B6. So if you lack B6, if you like, the homocysteine is shunted the other way. And if you lack B12, it's stuck. It's got nowhere to go. So it accumulates in the body. That's the problem. And that's why we need B6, B12, and folate particularly. Now, here's an old study in 1998. And what it shows is that the odds of developing brain shrinkage is remarkably high if you are low in vitamin B12. It goes up by, nearly 4.5 times. So, low B12, low folate, high homocysteine, all of these are a massive risk for increased brain shrinkage. Here is a study which looked at the change in homocysteine over 6 years and memory scores. And basically, if the homocysteine was going up, the memory was going down. If the homocysteine was going down, the memory was going up. And here's a study that came out this year. And those who look at science, we look for the odds ratio. So 10.475 means that those with high homocysteine are 10 times more likely to develop cognitive deficits. Now, 10 is a very big ratio to find in the study. But 17, I mean, this is very rarely heard of. A person with a high homocysteine in the study is 17 times more likely to have small blood vessel disease, cerebrovascular disease, which is what vascular dementia is all about. So homocysteine is right there in the middle of it. And the reason is that when homocysteine accumulates, for example, because you lack B12, it actually damages the arteries. It's a direct cause of heart disease. And 3 in 4 patients who have heart attacks do not have high cholesterol. 2/3 of heart attacks in the elderly are predicted by high homocysteine. It should be measured as often as cholesterol. So, it's nonsense that cholesterol causes heart disease. It doesn't. Cholesterol is a perfectly normal substance. And even the so-called bad LDL is not really bad. But what happens is it gets damaged by sugar. We've all heard of HbA1c, glycosylated hemoglobin, damaged red blood cells. It's how you, check for diabetes. And the cholesterol gets sugar damaged. The cholesterol gets oxidized. So if you smoke, you get oxidized cholesterol. If you eat too much sugar, glycated cholesterol, now it doesn't look right. And the immune cells that roam in your blood and in between, by the way, the top third is the bloodstream and underneath is within the arteries. It's on the lookout for something that doesn't look right, like a cancer cell or a virus or a bacteria. And it sees these abnormal cholesterol, damaged cholesterol, gobbles them up. And that vacuum cleaner bag of the immune cell, now full of damaged cholesterol, is called a foam cell. And a foam cell will lodge in the areas of the arteries that have been damaged by homocysteine. That's kind of how it works. So if you lower your homocysteine with B vitamins, you stabilize your blood sugar with a low glycemic load diet, less sugar and carbs, you have plenty of antioxidants, particularly vitamin C, And you also have omega-3s, which keep your blood thin, and magnesium, which relaxes your arteries. You're going to reverse this process. So what I'm showing you here is actually one of the most powerful pieces of discovery of what is driving most of the 21st century endemic diseases. And I could call it the 4 drivers of the health apocalypse. One is a loss of blood sugar control. The one is a breakdown in brain fats, omega-3s, phospholipids, vitamin D. The next is the increase in homocysteine due to a lack of B vitamins. And the fourth is too many oxidants, not enough antioxidants from fruit and veg. And they lead not only to brain cell death, but, death of arteries and so on. So, if I had 30 seconds to deliver a message to everyone, it would be no sugar, lots of omega-3 DHA, which means eating fish and seafood. If you're vegan, you've got to supplement it. It can be made from seaweed, from algae. So you can buy DHA supplements from algae if you're vegan. B6, B12, folate. Again, if you're vegan, you've got to supplement B12, and lots of veg and fruit. And the effect
Patrick,
of this, yes.
Can I just ask you, someone said that they've read somewhere that omega-3, high levels of omega-3 might cause atrial fibrillation. Have you come across that suggestion?
No, I haven't, actually.
I didn't think you would have. It does sound a lot like a theory to stop people eating healthily.
I think, no, I mean, atrial fibrillation, one of the things that's very interesting as a slight detour is that the heart cannot, I mean, the amazing thing about us humans, and we could do a whole lecture on this as well, is that we have the ability to run on either glucose, sugar, or ketones. Ketones are made in the liver from fat. And that's why some people eat high-fat, very low-carb ketogenic diets. Now, the heart muscle, that's what it is, a muscle, cannot only run on glucose, because if your blood glucose level, dropped massively, your heart would stop working. So it surrounds itself with fats, and it has the ability to make ketones very fast as an alternative fuel. And that ability to burn fats for the heart is done by an amino acid called carnitine. And carnitine and also coenzyme Q10 are absolutely brilliant at reducing AF, atrial fibrillation. And magnesium is also very important for the pulsing of nerves. So increasing magnesium, carnitine, coenzyme Q is superb for atrial fibrillation. Omega-3 is not really relevant. I'll continue. So here's a guy, I meet him in South Africa. He comes up to me in a seminar and says that he feels completely brain dead. He keeps losing his car in a multi-story car park. He's 59 years old. Really, he's in a mess. And we test him, and his homocysteine score is 119. And I tell him what to do, which included supplementing B vitamins, but also eating more greens and beans, high in folate, eating some seafood, getting some omegas and so on. And then when we retest him, after 3 months, his homocysteine has dropped to 19, and after 12 months, it's dropped to 9. And his memory, he says, is better than it has ever been before. So the point is, you can change these things quite quickly. So I want to start going closer, to B12 here, because there's a number of perfect storm factors that are contributing to this. And one is the very dirty drug, which are called proton pump inhibitors, which stop the stomach making stomach acid. I'll explain why this is completely the wrong way to go if you have gastrointestinal reflux disorder. And here is a recent study, because what these drugs do is they stop you absorbing B12, because if you don't have stomach secretions, you can't absorb B12. So it's not really a surprise, but it should be quite shocking that this study, as many others, shows that 4.4 years of use of proton pump inhibitors, like omeprazole, increases risk of developing dementia by 33%. So, that's quite a big association. Now, of course, so many people now have diabetes. What is it? 1 in 6 over 40, something like that. And the best drug, and it is, really quite a good drug in this context, is metformin. But metformin also interferes with the ability to absorb B12. So here's a recent study. It said metformin could be a major factor related to B12 deficiency, whereas concurrent supplementation of multivitamins may potentially protect against this deficiency. Serum homocysteine levels were negatively correlated with B12. In other words, if B12 is up, homocysteine is down. If homocysteine is up, B12 is down, suggesting that B12 deficiency due to metformin may occur at a tissue level, because if your homocysteine is raised, you're not doing methylation. Diuretics, which are used for high blood pressure. Again, here is an older study. There's not so many recent studies on this, but chronic diuretic use raises homocysteine, a significant decrease in the ability to retain folate, and no significant change in the concentrations of B6 or B12 in this case. Coffee raises homocysteine. So here's a study where having, if you look at the second paragraph, 2 cups of regular coffee raises homocysteine by 11% after only 4 hours, while caffeine tablets without coffee increase it by 5%. So it's not just the caffeine in coffee. There's something about coffee that raises homocysteine. If you have 1 a day, maybe it's not a big deal. But if you're drinking 5 or 6 cups of coffee, you're actually flushing B vitamins out of your system, including B12. And then, of course, what happens, you see your doctor and they measure your serum B12, which is the most common test, but it's quite inaccurate because most of the B12 in your bloodstream is bound and unavailable. What you want to measure is the free B12, which is actually measured by something called holotranscobalamin, or HTC. The next best test is MMA, or methylmalonic acid. So serum is the standard test. It's useful to have, but it's not nearly as accurate as HTC. It's a good starting point. The problem is that if you live in Europe or Japan, anything below 500 picogram per mil is considered deficient. But in the UK, anything below 180 picogram is considered deficient. So, so it's really a massive difference. In the States, the sort of bottom end of the reference range is 200. And this is just wrong. The science does not support those. You want to have a level above 500 units. And it's very clear from the evidence that you have accelerated brain shrinkage in that zone between the so-called OK level in the UK of 180 and up to 500. And the amount of B12 you need is literally whatever normalizes your homocysteine, which is saying that you are doing methylation. So if your homocysteine is raised, you're either not getting enough B12, folate, or B6, and we have to find out why. Now, this is probably one of the most important papers on homocysteine in the Journal of Internal Medicine in 2021 by Professors David Smith and Helga Refson. And if I read the first sentence, it says, we have reviewed the literature and have identified more than 100 diseases or conditions that are associated with raised concentration of plasma total homocysteine. There is no other marker, not glucose, not cholesterol, not iron, that has such a strong link with so many diseases. And at the bottom of that first paragraph, 5 diseases can at least in part be prevented by lowering total homocysteine: neural tube defects, impaired childhood cognition, macular degeneration, primary stroke, and cognitive impairment in the elderly. We conclude that you're safe probably below 10 micromole per liter, but really above 11, you've got to do something. In other words, supplement. And I won't go through all the diseases, but we'll see so many diseases that are also thought of and associated with B12. And of course, whether it's depression or bipolar or multiple sclerosis or Parkinson's, mental conditions, psychological, neurological, OCD, PTSD, schizophrenia, obviously cognitive decline and so on. I mean, really any neurological disease, any psychological disease, you've got to be thinking homocysteine and B12. So then the next question is, okay, so, are people really deficient in B12? And the answer is, according to a UK study, is that 2 in 5 people over 61 have insufficient B12 in their bloodstream to prevent accelerated brain shrinkage. So if you've got, 5 6-year-olds, 2 of them are in the brain shrinking zone. And then another study here looked at, took people who were mildly B12 deficient and gave them varying doses to see how much B12 supplementary was required to normalize their B12. And it concluded only doses of 647 to 1,032 micrograms was sufficient. Now, the RDA, or the ridiculous dietary arbitrary, as I call it, is 2.5 micrograms. So they had to give 250 times the RDA. And you cannot eat that. I mean, really, you would struggle to get more than 10 micrograms. So this is a situation where supplementation is absolutely essential. And of course, we go, why? And the answer is not fully unknown, but it's absorption. And it's to do with stomach secretions. Now, I used to teach this when I was teaching my nutrition students. I said supplements or food. And this was a study actually funded by the Food Standards Agency, which gave a third of the participants an amount of folate in broccoli, a measured amount in broccoli, the same amount in what we would call a yeast. They fed the yeasts. So we call it food state. You basically feed the yeast the B vitamin, it incorporates it into the food. And the third were given the cheapest, simple synthetic folic acid. And the question is, which worked better? And, we all love food and we want to get as much as we can from food as possible. But actually, it was the cheap synthetic folic acid was 1.7 times more effective in raising folate and lowering homocysteine than the food that contained the same amount of folate. So that was, I always sort of put that one in there. And here was a study I saw, saying you've got to have this food state, supplement where the yeast is fed the B vitamins and incorporates into the food structure. And, I looked at the only study done on this that was claiming benefit. And one of those lines, so this is hours after ingestion, increase in blood B12. One of those lines was the food state, and the other one was the synthetic folic acid. And if you had to guess which one it was, you probably couldn't, because they're kind of similar. One starts higher, ends up lower, et cetera, et cetera. So there really is no evidence of that sort of food state really being better. Then, I'm always a little bit skeptical, but, it is true that ultimately from folic acid and also from folate in food, we have to generate, and I said the nutrients you eat don't work, they have to be processed into something. And all those green, there are stages where we turn that folic acid or the dietary folate into something called 5-MTHF. And we generally call this methyl folate. So folate with a methyl group. And some people are particularly bad at doing this because they lack an effective gene instruction, so that they're not so great at doing it. And there became a little bit of a concern around this because we knew that while folic acid, this is the cheap synthetic version of folate, the B vitamin, is very strongly linked to low cancer rates. If you feed precancerous cells, like, for example, colorectal polyps as precancerous cells, extra folic acid, the cancer cell will grow. So generally anti-cancer, but actually can stimulate the growth of cancer cells, which is not so surprising because cancer cells are trying to survive and, nutrients are sort of important. But really what happened was that commercially, two companies, Merck and one other, were able to find a way to make a methyl folate, and it's a patentable product. So they put a lot of money into promoting the idea that all folic acid is bad and you have to have our methyl folate, which is better. And it probably is safer, but it's not massively better. In other words, in this study, what you're seeing is that the methyl folate was versus folic acid for 24 weeks, lowered homocysteine by 14.6% versus 9.3%. So a little bit better, but not a lot better. And this relates specifically to people who happen to have a gene polymorphism of the MTHF gene called C67TT, which affects about 25% of people. So yeah, methyl folate is better, but don't throw the baby out with the bathwater, because so much research has been done on folic acid and it has worked. So the cofactor nutrients that are required for methylation that lower homocysteine are folic acid, folate, methylfolate, B12. Some people give methylated B12, methyl B12, B6. Again, B6 itself doesn't work, but when you have enough zinc, you make pyridoxal 5-phosphate or P5P, which works a bit better. B2, B3, niacin, and trimethylglycine. In the liver, there is a pathway for processing homocysteine that depends upon zinc and trimethylglycine, which is very rich in sugar beet, by the way. It's called betaine, is the other name for it. So betaine hydrochloride, stomach acid. Betaine is trimethylglycine, an amino acid with 3 methyl groups. We, of course, take the sugar out, the sugar beet, feed it to humans, we get fat and sick, and the animals eat the rest and they stay healthy. And then that pathway leading towards glutathione. If you have enough N-acetylcysteine, you satisfy that pathway, you shunt the homocysteine towards SAMe, which does the methylation. So homocysteine, on average, goes up with age. It doesn't mean it's good, but it just does. And I'm 65, so the average person at my age is going to have a homocysteine level of about 12, which we've already learned is enough to be causing accelerated brain shrinkage. The question is, what is the optimum level of homocysteine for optimum methylation is debatable. But we know below 7, you have the lowest risk of coronary artery disease. Below 7.3, actually, above 7.3, you get chromosomal damage. Below 7 means the best psychological function. Above 9, pregnant women who have a homocysteine above 9 have children aged 2, 4, 6, 8, and 10 who have more psychological issues. Above 9.5, or maybe we should say 11.5 here, we have clear evidence of accelerated brain shrinkage. Professor David Smith and Helga Refsum recommend action when your level is above 10. And what they gave in their study was 20 milligrams of B6, 500 microgram of B12, and 800 microgram of folic acid. But nowadays, they think it would be better to give less folic acid, maybe 400 microgram is sufficient, and in the form of methylfolate. Now, there really are not the studies, also including B2 and zinc and TMG, trimethylglycine, NAC or glutathione, but it's very logical that a lot of homocysteine-lowering formulas contain all of those nutrients. But this is the key is you've got to have 20 milligrams of B6, 500 microgram of B12. And generally, in my experience, but you'll know a lot more than I do about this, the majority of people who have a problem with B12 and pernicious anemia can raise their B12 and lower their homocysteine by high supplemental B12. Some may need more than 500 micrograms, maybe 1,000 micrograms. Some, however, it just doesn't work. They've got to have, injected B12 at whatever frequency works for them. And maybe even for them, there's a sort of middle ground where you could, have a lesser frequency of injection, provided you also supplement in between. So I remember many years ago formulating a supplement called Connect, which provides all these nutrients. And then I helped other companies. Higher Nature made one called H Factor. Solgar have one called Homocysteine Modulators. Cytoplan have one, and so on. But generally, and on our website at the charity, you can actually go into the area that says books and tests and supplements, and you can look at a list of those supplements and what they provide in relation to lowering homocysteine. But, usually you're taking 2 a day, and it will normalize homocysteine. So now we have to understand, well, why are we not absorbing it? So the stomach is an acid bath, and that acid has effectively 4 functions. So when you eat protein, your stomach makes stomach acid. Stomach acid is betaine hydrochloride. The betaine part we've learned is trimethylglycine. So it's some nutrient that's very rich in fish, and it's rich in root vegetables and all sorts of foods that contain TMG. The hydrochloride part is actually made by a zinc-dependent enzyme. So if you were lacking B vitamins and couldn't do methylation, you wouldn't have the trimethylglycine part. And if you're lacking zinc, you couldn't have the hydrochloric acid part. So already you have a problem. And that, when you eat protein, your body produces stomach acid, which shuts the valves at the top and bottom of the stomach, creates an acid bath, which has a couple of effects. Number one, it digests your food. Number two is it kills your bugs. So it's like the sanitizing department. So while you're breaking down protein, which takes 2 to 3 hours, all the bugs are being killed. It's a bit like making compost at a high heat. You know, it kills the seeds. And also, your stomach makes intrinsic factor. And intrinsic factor is required for B12 absorption. So that's kind of what's going on. Now, if you lack stomach acid, and by the way, there is evidence, not enough evidence, not enough studies, I think, on this, that as you get older, your amount of stomach secretion produced could very easily become much less. The effect of less stomach acid, if you think about it, is on the one hand, you can't digest your food properly. You can't kill off bugs. So you get the wrong bugs in your microbiome, and the bugs eat the food you don't digest. So you get gas, which makes you bloat, and the gas has to go somewhere, and it's high up in the digestive tract. So you belch, and you haven't shut the valves in your stomach. So you're going to belch a bit of stomach acid. That will get into your esophagus. That will give you heartburn. So you go to your doctor with heartburn, indigestion, bloating, and belching, and they're going to give you a drug that stops you making any stomach acid. And of course, the heartburn will go away because no stomach acid, no heartburn. But we generally, provided we've always checked that you haven't actually got a stomach ulcer in the first case, we will give you the opposite. We will give you stomach acid. You can literally get supplements of HCl, betaine hydrochloride, and pepsin. And so often people who have these problems where they belch and get bloated after protein, they take the supplement and it stops instantly. You know, it's just a pure piece of dynamic. But of course, it's not patentable, it's not profitable. And if you think about it, you know, the wonderful New Zealand doctor who gave himself Helicobacter pylori infection and proved that it caused stomach ulcers, why would one have a bacterial infection in the stomach? And the answer, I believe, this has not really been substantiated, but based on the logic, is lack of stomach acid. So now we come to the hunch of what happened in 2010. There I was with Professor David Smith. He'd set up a randomized placebo-controlled trial. Half of the people with pre-dementia, MCI, were given B vitamins, half were given placebo. And right here, we're looking at the placebo group. They were split into their homocysteine level from the lowest quarter to the highest quarter at the beginning of the study. And then their rate of brain shrinkage was monitored from the beginning of the year to the end of the year. And what you're seeing here is the higher the homocysteine was, the greater was the rate of brain shrinkage. So homocysteine predicts your rate of brain shrinkage. Those who were given the B vitamins had much less brain shrinkage. And it became especially obvious in the quarter who started with raised homocysteine, 53% lower rate of brain shrinkage. And to make this real, on the left, you see someone who has a homocysteine level of 22. By the end of the year, it's risen to 30. Their total brain has shrunk by 2.5%. The areas in turquoise are the areas of the most shrinkage in the central part of the brain. That gap in the middle is the top of the spinal cord. They're on the placebo group. The person on the right, luckily, was in the B vitamin group. Their total brain shrinkage is less than 0.5%, which is quite normal without measuring any loss of cognitive function. There is no turquoise, there's no visible loss of brain matter, and their homocysteine has gone down from 24 to 12. Here is going close up on the Alzheimer's areas of the brain, and what we're looking at is 9 times less shrinkage in the Alzheimer's areas of the brain in those given B vitamins, which included 500 microgram of B12, which is probably the single most important, followed by the folate and then the B6. So as we saw earlier, those omega-3 fats, mainly DHA, bind to phospholipids. One of the most prevalent phospholipids is called phosphatidylcholine, very rich in eggs. I'm a great fan of eggs. If you don't eat eggs and you don't eat, meat or fish, then lecithin granules is a vegan supply of phosphatidylcholine. And those black and white things are actually cholesterol. 25% of the body's cholesterol is in the brain. It's a sort of filler. It holds things together. So if you actually look at the blood tests that most predict dementia, 1, raised homocysteine, 2, low cholesterol. A cholesterol below 4 is an extremely good predictor of cognitive decline. You do not want to drive your cholesterol too low with medication. So attaching omega-3 to phospholipids requires methylation. And realizing that, some years later, Professor David Smith and his group with a scientist called Frederick Jernerin went back to their original blood samples. They didn't give omega-3, but they looked at the omega-3 level in the blood at baseline at the beginning of the study and split them into the lowest group on the left and the highest group on the right. And what they found is that those in the lowest third of omega-3 had no benefit from the B vitamins. B vitamins do not work without omega-3. Now, that's a really important thing for someone who has a B12 problem to realize, is the B12 doesn't work in its main function, which is methylation, if you do not have sufficient omega-3. But those who are in the highest third, not necessarily optimum, just the highest third, had not 53%, which was the average shrinkage reduction, but 73% lower rate of brain shrinkage, which brought the rate of brain shrinkage down to that seen in perfectly normal people. And also the clinical dementia rating, which is a measure that's being used on all these new anti-amyloid drugs, none of which have made a single person better, just marginally less worse than those on placebo. But in this trial, 30% ended the trial with a clinical dementia rating of 0. That means they would no longer be clinically diagnosable as having dementia. They have no brain shrinkage that would classify them as dementia. So in other words, every indicator of dementia has ceased. This is gold. And realizing this, another trial, which had a Swedish trial that had given omega-3, a very decent dose, 2.3 grams, like 2 big fish oil capsules, and got no significant effect overall. They went back to their original blood samples and split the group into the third with the lowest homocysteine and the third with the highest homocysteine. So on the right, you see the highest homocysteine above 15.7, no effect of the omega-3 versus placebo. But the third with the lowest homocysteine, below 11.7, had a massively statistically significant clinical effect, about 3 times greater than any of these anti-amyloid drugs. So again, the omega-3s do not work without methylation, which means B vitamins. And then another trial, this was a Dutch trial, a very good trial, had given B vitamins and found a marginal benefit. I mean, it was statistically significant, but not, massive. And they thought maybe we're missing a trick too. So they went back to their original blood samples and split the group into thirds. And the third with the lowest omega-3 DHA, no benefit from the B vitamins. But the third with the highest omega-3 DHA, massive clinical improvement. So we really know now that these two are codependent. The homocysteine-lowering B vitamins, B12, folate, B6 require omega-3 to have their effect in the brain. So how common is it to have a homocysteine level higher than 11? Well, 40% over 60 in the US are above 11. In China, a recent study, the mean homocysteine levels in adult males less than 30 years of age and greater than 60 were higher than the upper limit of normal, which is 15 in their sort of language. And in the UK, we don't have a really good survey, I have to say, but 2 in 5 over 61 have insufficient plasma B12. So it's a lot of people over 60 can be expected to have a high homocysteine. And we think that this is causal. It's causal because when you increase the B vitamins, especially B12, you lower homocysteine. When you lower homocysteine, it prevents that regional gray matter atrophy. It improves clinical dementia ratings. It slows cognitive decline. And all of this is completely consistent with a modification of the actual disease process, while the anti-amyloid drugs are not, they're not causal. There is no evidence that they are causal in this way. So if we just take a look, up until recently, the anti-amyloid drugs claimed to have reduced brain shrinkage by around 4%. B vitamins on their own, 52%. B vitamins and omega-3 DHA, 73%. But actually, the latest trial of donanemab had 20% increased rate of brain shrinkage. Now, I read the paper, and it's right there in black and white. It's a very obvious thing to look for, not mentioned in a single newspaper coverage by the BBC or the Guardian or anybody else, just simply not mentioned. So here we have one treatment with B vitamins and sufficient omega-3, 73% less brain shrinkage, and another treatment with 20% increased rate of brain shrinkage. Which one would you have? In relation to clinical dementia reduction, so that's a sort of, it's actually done on a questionnaire, and you can cheat on this. I mean, I'm afraid to say that, you know, this is what happens. Never trust fully a study on vitamins done by a vitamin company or by drugs done by the drug company. You know, what you want is an independent agency, you know, to do the study. And we've seen this with statins. There is not a single independent study on statins that have shown benefit, but the studies done by the makers of the statins have shown benefit. And the way it works is you actually ask the carer or the partner, you know, do you think this person has got better? And of course, they're in a drug trial. They're not meant to know they're on, you know, placebo, but actually what happened in this last one is there were 3 deaths, 20% got brain bleeding, 24% got brain swelling. In the previous one, there were 2 deaths and a third got brain bleeding or swelling. So, you know, it's quite easy to guess that you're on the drug. And, you know, you've got a lot at stake and you're really hoping your beloved partner, you know, is going to benefit. And when you're asked a question, do you think they're functioning a bit better? You say, well, yeah, actually, I think I'm going to, you know, mark them up a point. And, you know, and then you get an effect. In truth, this is just playing around with statistics, because the only thing that counts is when you get to a clinical dementia rating of 0. You know, being marginally less worse, it doesn't mean anything. It won't mean anything to the patient or the carer. It's really ineffective. So in my view, the amyloid, you know, theory is wrong. It doesn't work. And I'm not the only one. You know, there's some lovely reviews in the British Medical Journal and so on. So then the next big focus has been on abnormal neurofibrillary tangles. So the amyloid produces plaques, and the tau protein produces these tangles. So it's like those, you know, those nerves signals are all sort of tangled up and messed up. And the body actually makes the tau protein. It turns into the less desirable phosphorylated tau, and then it's turned back into tau. And really, you want more tau than p-tau. And when you got more p-tau than tau, then that predicts neurofibrillary tangles. And guess what? Homocysteine blocks the enzyme that gets rid of the bad stuff, and it activates the enzyme, you know, that turns the good stuff into the bad stuff. So if you want to lower p-tau, what you do is lower homocysteine. And how do you lower homocysteine? With B vitamins. So, you know, the big story is don't forget your vitamins and your omega-3 DHA. They're absolutely vital to the brain. So what we've been doing at the charity Food for the Brain is encouraging as many people as possible to go online, do the cognitive function test, which is followed by a questionnaire, which then grades your risk across these 8 domains. So you get 3 pieces of information. You get your actual cognitive function score, which you want in the green. So that sort of lower zone of orange going to red is really dementia. And the way we've devised the questionnaire, even though we'll ask you about things like head trauma, early education, ApoE4, we don't use a score for them because you can't change them. So if you're doing everything wrong in your diet and lifestyle, you score 100%. as your dementia risk index. And if you're doing everything right, you score 0%. So you have under your control the ability to drive your total risk down to 0. And it shows you those 8 domains, and it shows you what's driving it. So for this person, low GL and carbs, so it's their sugar and carb intake that is their single greatest risk. But they've got an active mind. That's not a problem. You know, they're using their mind. So it shows you where to focus. And on our website, you can click on cognitive function test. Please join as a friend. It's £50 a year. Everything we've done is just simply by people joining as a friend. Read the manifesto. If you're into this, there's an amazing masterclass. I think it's only £15 of all the experts that I've introduced you to. Go to the For You section, click on Prevent Alzheimer's, lots of great articles, films, 3 films. I showed you one on how to build a brain. Find out what your score is. And then this is a really nice piece of gold, because really this is for total health. If you control your blood sugar with a low glycemic load diet, up your omegas, vitamin D, phospholipids, eggs, homocysteine low, plenty of antioxidants, you're really not going to get sick. Now that blood sugar, but you say, oh yeah, I'm absolutely fine. Why don't you ask your body? If you measure your HbA1c in your bloodstream, it actually tells you where you are on that scale of sugar control. Oh yeah, I get enough omega-3, absolutely. And yeah, vitamin D is fine. Measure it. Find out in your body what's going on. Homocysteine, measure it. And I'll tell you something I've been working on for the last few months, which I hope will come into fruition about 6 months, is the single best measure of your total antioxidant power, so to speak, is something called red cell glutathione. That's the most potent antioxidant. And I've worked out that the loaded form of glutathione, which is called reduced, divided by the spent form of glutathione called oxidized, is the best, the ratio. And we hope to launch all of these as a single pinprick blood test. And I'll tell you, for those of you who have been following the action, we are a tiny charity. There's 4 of us working 2 days a week, apparently, doing all this, and then a team of amazing and wonderful skilled volunteers supporting it. It's extraordinary what you can do with goodwill and digital technology. So you will be able to buy, by the middle of December, a kit to measure your homocysteine. So homocysteine is coming middle of December, the kit. But we won't actually start analyzing until the new year. So you can buy the kit and maybe give it to someone for a Christmas present, but don't open the kit and prick your finger and register and send it in until the new year. So, and also you'll be able to buy a kit, a single kit with a strip with a place for 4 blood spots. So you prick your finger. It's a very, very neat system. Doesn't hurt. It's very clear. 4 drops of blood, and it will measure your omega-3 status, your vitamin D status, your homocysteine status, and your HbA1c. And secretly, in about 6 months, I hope to add glutathione. And the cost of all that is under £180. And we're trying to keep this as cheap as we possibly can. And this is available across the EU and the UK. Not yet. All of these are available in North America except the homocysteine. We hope by the middle of next year to have the homocysteine available there too. So we want you to get involved. Do the test, not just for yourself, but because you're adding to this whole research database. Fill in the questionnaire, find out what's driving your risk, and hopefully change it. And we encourage you every 6 months to come back and redo the test. And we are therefore tracking what happens to those who do it or not. And you have the option to join something called Cognition. And I'm just about to knowing what you should.
Another thing, doing it. That's what signing up for Cognition is all about. We specialize in helping you to change one step at a time until all your areas of risk are in the green. How? You pick one of your weakest domains. Then every other day you'll get an assignment, something to read, watch, or put into action. You'll also receive motivational prompts by text to motivate you and keep you on track. And optional Zoom groups to join with our health coaches, webinars to watch, and full support via the Friends Facebook group, our community of people like you committed to making changes to dementia-proof your diet and lifestyle. At the end of each month, you reassess your domain, then move on to change the next until you end up with all in the green, with your dementia risk index dropping close to 0. We even offer you pinprick blood tests to confirm you've optimized your intake of B vitamins, omega-3, vitamin D.
So that's what we've been up to. And that £50 a year friend gives you access to all this personalized education. I'm going to end with the story of Alan Norris, nicknamed Nodge. On the 15th of December, he's diagnosed with mixed dementia, Alzheimer's and vascular. You can see his cognitive function score is not good. And he already knew quite a lot. You know, he took omega-3, but didn't really know about the B vitamin thing as well. And his lovely wife, Dorothy, ran him through the test. They found our charity and they started to do everything. He went to bed an hour earlier to get more sleep. He started to have something called C8 oil or keto fast. It makes ketones, very good for the brain. He went 5 days, he went ketogenic a month, 5 days a month, full on ketogenic. Cut right back on sugar, supplemented the B vitamins, high dose B12, plus omega-3s, and even joined a Morris dancing group. Now, Nodge, his wife kept finding him in a different room at night. He couldn't find the loo in his own house. He was a computer programmer. He couldn't turn his computer on or off. He was a gardener. He couldn't even talk and eat. He couldn't think of tomorrow. I mean, the concept of tomorrow, it really was not there. And 3 months later, 14th of March, his dementia risk index has dropped from 56 to 43%. His cognition has improved substantially. He's completely planted his garden out. He's joined a Morris dancing group. And Dorothy Dot says, I've got my husband back from dementia. So, we cannot claim anything, and that's not appropriate to do so, but we really think this is a preventable disease for the majority, maybe 99%. Please do the cognitive function test at foodforthebrain.org. Please tell all your friends to do it. It'll help to put B12 on the map, because it is such a vital piece of this. And if you need to contact me because you have groups or, particular things, please not just individual questions. My name is Patrick. My email is patrick@foodforthebrain.org. And finally, B12d, your organization can, there's no cost for this, say, hey, we would like to look at our whole group. And we'll give you a URL. You only have to ask us. It could be foodforthebrain.org/B12d. And everyone who comes in and does the cognitive function test is tagged to your group. The individual results are anonymized immediately, so there's no chance of your email or anything being shared. But you can actually then see your whole group, everyone who's done it under B12d, where you start and where you change over time. So as a group, you can kind of encourage each other to make changes and see if you can't shift, the whole group to the point where you're all in the green. And that's how you get involved in being citizen scientists. And finally, we've employed the brilliant services of Dr. Christina Curtis. She runs the Applied Behavior Change Unit, and she's coming into each of those 8 units, those 8 domains, to really research how to change people's behavior. She's been incredibly generous to us. We have to raise £85,000 to do this whole study that actually leads to Mark II, which will be the most effective we can get to in changing behavior. And all of this, as I said, is funded by people becoming a friend of the charity. It's only £50 a year. It makes such a big difference. And, this is science done by the people, for the people, and everything we do is shared right back to you. So last slide, how to dementia-proof yourself. Take the test. Find out what's driving it for you. Up your intake of omega-3, eat oily fish, seafood, supplement as well. I do both. If you don't eat any fish, you've got to supplement. Check your homocysteine. If it's raised, supplement B12, B6, folate. at the right doses, cut back on carbs and sugar, eat more greens and berries and nuts and seeds and beans, which are very rich in folate, drink less. Alcohol, I actually should have had a slide on that, robs the body of B vitamins massively. But 125 ml, a small glass of red wine actually is less risk than nothing. Active lifestyle, physical, social, intellectual, reduce stress, sleep well, get outdoors, and check your vitamin D level, especially now we're in the winter. You want it above 75 nanomoles per liter. Thank you very much.
Thank you very much, Patrick. That's been terrific. And we have had a huge number of questions. Well, a number of questions, I should say, some of which I've answered already. But I will nonetheless repeat them for you. So someone asked, is water fasting a good way to reduce homocysteine?
No, it's not, because, we've seen that homocysteine accumulates when you have a lack of B vitamins. So, if you water fast and you have no B vitamins at all, then how are you going to get rid of the homocysteine? So, no.
Absolutely. And that was the next question is there a B6 toxicity if you take too much of it, given its importance in reducing homocysteine?
Not really. I mean, there's a big myth around B6. And you know how I said earlier that a lot of the nutrients we take in don't actually work. They've got to be acted on. And this is true with B6. So B6 has to turn into pyridoxal 5-phosphate, which requires zinc to do it. And then there's actually a methylation process required as well. So there have been a couple of cases of people who were given B6, pyridoxine, at a very high level, 1,000 milligram, who developed peripheral neuropathy, that is tingling in the extremities. It has never, ever occurred when someone was given B6 and zinc. And it has never, ever occurred in someone given pyridoxal 5-phosphate. So actually what's happening here is the very high dose B6 in the absence of cofactor nutrients like zinc blocks the lock that the key has to open up. So in other words, it creates B6 deficiency, in effect, the very high dose B6. So this peripheral neuropathy, you people into B12, you know well about, because it's probably one of the earliest, classic warning signs. It's definitely not the earliest sign, because things like depression and tiredness are probably the earliest experienceable symptom. But once you've got peripheral neuropathy and tingling and numbness and strange things going on at the extremities in relation to your nerves, you're starting to think something's wrong. So B6 itself is not toxic, but if it's given on its own in a very large amount, it could be. But really, up to, I would say, at least 200 milligram, you've not got a problem. We were talking about 20 milligrams. 50 is really not a problem. 100 is not a problem. So I wouldn't worry about it, basically. I mean, here's an extraordinary thing. And I was just interviewing a psychiatrist the other day who only uses natural medicine. She's part retired. She's in California. I said, how much is your insurance? And she said $3,000 a year. But she says, my insurance is the lowest of any, psychiatrist. And she said, how much is your insurance? And I said, well, I used to think as a nutritional therapist, it was £50. But I've since learned through disclosure, it's actually £25. The remaining 25 is part profit and part admin. So I've got a £2 million cover for £25 a year, because basically since 1981, when I wrote my first book and have been extolling the benefits of nutrients at the levels that the science supports, I have not had one single communication to say that my disease got significantly worse. So your body loves these nutrients. It's part of its evolution. And your body knows exactly what to do with them. And B12 is a classic example. It's phenomenally non-toxic. RDA 2.5 microgram. I'm sure there are many of you who supplement 1,000 microgram. Is there any evidence of toxicity of that? Not that I'm aware of.
I joke that the only toxicity you can get from B12 is if a large box falls from a high shelf.
It's true.
There's a lot of enthusiasm. The most popular question that we're getting is, where do we get tests for homocysteine? Where does it appear on the test sheet? And how do you persuade someone to do your test? Can you even do a test outside hospital?
Yeah. Well, I think it's almost, it's so weird that it's been so hard to get it tested. One almost, I mean, I will tell you that one of the big pharma guys came to Professor David Smith and said, if this was patentable, this is a multi-billion-dollar treatment. But it's not, so we don't want to know. And no one denies this evidence. There's no, rebuttal of it. The evidence is as solid as a rock. It's just ignored. So any doctor can get a homocysteine test done. There is something about homocysteine that makes it slightly trickier, which is in the plasma, which is the clear part of your blood. So the conventional way of testing is that you'd go to a lab, they'd take a blood sample, a blood draw, they'd spin the blood to get the clear part, test it there and then, or freeze it, or fix it, and then test it. So it's a little bit more complicated. Like cholesterol, you can just take the whole blood and, it's relatively stable. So I've been committed to getting homocysteine available, and I've been working with some very brilliant scientists using what we call dry blood spot technique, where we don't have to separate the blood into plasma. We use the whole dry blood spot, and we have a perfect correlation with plasma. So it's a slightly different actual measurement, so we have to multiply by a factor. And we actually ran one validation study on 73 people. And there's another one we've just completed on 123. This is quite big now. There are 4 other groups who've done the same thing. And I really wanted to get to the point where we could categorically say that what we're measuring, produces a plasma equivalent that is accurate to, within 10%. No false positives where you get a high score and you aren't, or false negatives. And because of that sort of 10% variation, I am more comfortable saying, if you are above 10, do something about it, even though the studies show 11, is really the cutoff point. And we will definitely be measuring homocysteine from the new year, but we will definitely have test kits that you can order by the middle of December. And it actually helps us. We actually have to buy and make all these test kits, and we're just a tiny charity. So if you do order a homocysteine, come back on December the 15th, or just get on our mailing list. If you go to the website and you go down to the bottom of the homepage, you can subscribe or become a friend or do the test. And when you do the test, you have to give us permission to use your data anonymized. So if you do that, we will let you know. We will let you know the second the homocysteine test available. The cheapest we could get it down to is just under £50. I think 40, there's certain, if you have it with other tests, it becomes a bit cheaper. I wish it could be even less than that, but at least we've broken this stranglehold on not being able to get the data. And I want to tell you a little story. I was at a lecture a few years ago, and there was a woman there with a stroke. Strokes are terribly strongly linked to homocysteine. Anyone who's had a stroke needs to measure the homocysteine. And stroke recovery is improved by lowering homocysteine. The mother had had a stroke, and the daughter said, "Mum, I really think you should have a homocysteine test." And the mother said, "I will if you will." The daughter, by the way, had chronic fatigue. The mother's homocysteine was 6, which is perfect. So her stroke had nothing to do with methylation or B vitamins. The daughter's level was 26, which is much too high. And she lowered her homocysteine over a period of a month with supplements and a few diet changes, and her chronic fatigue went. And then a few years ago, a dear friend of my wife collapses with leukemia, and she's kind of saved by, the chemo and the medical intervention. But the question then is, why? And this is really the way of thinking. Is it blood sugar? Is it the GL thing? No, she was fine. Is it brain fat? Is it fats? Is it omega-3? No, it was fine. She supplemented omega-3. Was it vitamin D? No, that was fine. Is it antioxidants? She grew her own veg. I measured her red cell glutathione. It was fine. Is it methylation? Her homocysteine was basically 40. Why? Well, she's a malabsorber of B12 and didn't know it. And if you don't absorb B12, you can't do methylation. If you can't do methylation, it scrambles your DNA. You can't repair your DNA. And the link between homocysteine and leukemia is massively strong. So you're thinking in that way, and you can suddenly find someone who has no reason to know that they're actually a malabsorber of B12. And that is why we have to test. And the best test is not B12 itself. The best test is homocysteine, because if the homocysteine is high, you are functionally not doing methylation. Now, you don't then know if it's B12 or folate, but you know you're not doing it. So then you start to explore and you find out, what is that intake of B12 that gets my homocysteine down to 7?
Yes, absolutely. And it's really good. And people are enthusiastic about the fact that you have a test now for homocysteine, or you will have a test for homocysteine that people can actually do that comes up with a good correlation. It is now past, so for those people who need to go, who are watching the clock, I know someone left us on the hour, but for those who are watching the clock, then go. I needed to tell you the time. I'm going to beg of Patrick's time for a few more minutes. Thank you. And I'd just like to remind everybody, we will issue the recording when it's been checked and when Patrick's approved it. And we will also issue the PDF of the slides when that's available. But do follow the link through to his website. I've put them into the chat to different parts of his website, and do sign up as a friend and follow through on the cognitive tests and help him do the research he's doing, which is really useful, going to be very powerful and very helpful to all people who are involved in suffering from B12 deficiency, or caring for people who suffer from B12 deficiency, or health professionals. The last couple of questions. People are saying that they're being denied access to homocysteine tests, including in hospitals. Is there reason to deny them access?
I think the problem is that, there was a completely inaccurate sort of downgrading of the link between cardiovascular disease and homocysteine. It's just wrong. So it used to be so that you could get a homocysteine if you had a compelling reason, and the compelling reason could be cardiovascular disease or risk, and it could be stroke, definitely is strongly linked, and it could be, cognitive decline. But for other causes, they'd say, we don't have the evidence, so we're not going to test it. It costs the doctor about 30, 35 pounds, something like that. So, for a doctor to run a test, they have to kind of have a reason to do it, and they don't see it. And the honest truth is they don't know, because they don't study it. They're not taught it. If you go into PubMed, which is where all the research is, it's about 30,000 studies on homocysteine. So I honestly think, to some extent, we may feel a lot better. It was old George Bernard Shaw who said, everyone complains about the weather and nobody does anything about it. I think the NHS is broken. It's the fastest growing failing business. And it's broken and will continue to cost more and function less, for the very simple reason that it isn't doing medicine. It's not doing prevention. And the only thing that will save the NHS is not more money, not more doctors, not more nurses. It's less patients. And the only way to get less patients is to have more prevention. So I would say, test yourself, take action, get on it, own your own health, and do everything that you can, because unfortunately right now there's not a lot of support. And I say, as someone who's been waiting, to get a test done, I think for 14 months now, So what I love, I had no reason to focus on dementia, by the way. I've written books on cancer and heart disease and all the rest of it. But I thought this is a perfect opportunity, because unlike diabetes, which is reversible, I got my head chopped off when I first said that and proved it. Because it's not reversible, but is preventable. And because it's the greatest fear and the greatest healthcare cost and almost the greatest killer, it's kind of, it's spiky. And in 2010, when I first started the Alzheimer's is Preventable campaign, people laughed at me. The idea that you could prevent Alzheimer's was a complete joke. But now there isn't a week you don't read the newspaper saying, if you do this or this or this, you reduce your risk. So I thought I would focus on this, because the honest truth is every single one of those risk factors is what's driving everything else. So then the question is, how do we do prevention on a major scale? How do we get hundreds of thousands, if not millions of people actually doing prevention? And this questionnaire and the test, anyone can do anywhere in the world. We're just having it translated into Japan. We hope to get it translated into many, many languages. And immediately it makes the invisible visible. And you go, my God, my dementia risk index is 50. You may be fine in your function right now, but you can see your future risk is something. And then you start to wake up and you start to learn and hopefully share that with others. So I think we have to stop thinking that the doctor's going to sort our problem out and start thinking it's my health, it's my body. And really, you know, we're only born with this body and this consciousness. We don't know where the consciousness comes from. It's there. It's always there. We don't know if it goes or not. You know, it's a miracle. And this body is a miracle. The human body is a miracle. And I was talking to a Swami who's staying at my farm in Wales. And she's amazing. Every day she's in her 80s. She's out there weeding and she loves nature and she's making nature really, really beautiful. But she smokes and doesn't eat very well. And I said, it's extraordinary, isn't it? That the closest nature, you know, we can love nature out there, but the closest nature is the nature of our own body. And, you know, you need to respect the body because it's through the body we have all the wonderful experiences of the senses. So your closest nature, if you believe in nature, is your body. You know, take respect for your body and it will pay you back 100-fold.
Another question that's been coming through is how much sugar do we need to cut out? Do we have to cut out all of it or can we just cut down a bit?
Yeah, well, there speaks addiction. You know, I've long since thought what is actually driving disease, and the answer is there are only two things. One is ignorance, and the other is addiction. Right now, you've got 65,000 miles of blood vessels inside your body, and there exists in that 65,000 miles 3.5 grams of sugar. So that's about 2/3 of a teaspoon of glucose is the total sugar in your entire body at this point in time. That is all you need. A can of Coke is 10. So I have not bought sugar for 45 years. You know, I just don't buy it. And nearly always I don't buy foods containing sugar. Now, I had a lovely Thai meal and obviously they could use a bit of sugar and so on. But basically sugar is just not kind of something that I use. And as a consequence of that, I mean, I used to have 3 teaspoons of sugar in my tea when I was a student. And now half a teaspoon would be really sweet. So as you reduce your desire for sweetness, you become used to much less. And the honest truth is that nature never puts sugar in a food without putting fiber. So you've got to eat your fruit, not drink it. And eat less carbs on the whole. And of course, if you're doing a lot of exercise, you can probably get away with more. So I would say turn the equation around the other way, which is have a week or 10 days with way less. Maybe get my book, the GL Diet Bible, GL Diet Cookbook. It's really, really practical. And just follow that. And if you are a bit of a sugar addict, you have a 3-day withdrawal process. So you may feel a bit worse for 3 days, but usually after a week, you start to really come back to life. And I have to say, I'm 65 years old, and there is only one significant problem that does occur if you do everything right in relation to nutrition, and that is you just have so much energy and mental clarity. You know, you wake up alert, things work, you know, ideas come together, and you know, I write books. You know, I've just finished a book. It's called Upgrade Your Brain. It will be out on April the 25th. So, you know, if you're struggling with energy, know that if you do it all right, it's going to make a phenomenal difference. And I remember on a post I did on COVID on Facebook, it was vaccines or whatever it was, but I love the comment. Someone said, "A shame there isn't a vaccine against stupidity." But I have to tell you quite seriously, if you look at the current, you know, we're down to 1 in 6 children with special educational needs right now. We're losing 7% of IQ a generation. And on the current trend, this is straight statistics, on the current trend, by 2080, 1 in 3 children will be labeled mentally retarded on our current demise in brain function. So not only do we need to get on this for ourselves and our family and so on, But if we don't really have a major paradigm shift and really wake up to what's going on, what we're looking at is the demise of humanity. And as you've probably noticed, our politicians are not so clever. We're just not making smart decisions. I mean, the guy in World War II who said, right, everyone needs a spoonful of cod liver oil, was really, really onto something. But we don't even have that thinking. So really, nutrition and particularly mental health and things like B12 have got to be at the very top of the health agenda. At the moment, we're hardly squeezing them in at the bottom, but they got to be at the top. There have been 3 government reports now, all of which have said the single biggest issue is mental health, single biggest cost is mental health. And as Darwin said, it's the conditions of existence, which we have changed beyond belief. We are not eating anything like the food that we evolved to eat. And, there lies the problem. We are literally digging our own graves with a knife and fork.
Which brings us round to the last question I'm going to ask, which is there any hope for elderly people we're looking after who have dementia?
Well, I'm quite glad you asked that, because actually, an interesting thing, somebody reminded me of this in I have a podcast at patrickholford.podbean, and I interviewed David Smith. And what we find is even late in the process, when you get someone's homocysteine down, B12 up, omegas and so on, they say you won't always stop the cognitive decline, but they get their personality back. It's a really interesting thing. So that can help. But if we have more time, I would talk about something very, very interesting. The consequence of too much sugar is you become insulin resistant. Insulin is the hormone that drives glucose into, among other, brain cells. So the consequence of too much sugar is you actually have sugar deficiency in the brain cell. And the consequence of having glucose deficiency in the brain cells is that you're mentally tired and consequently can't remember and can't work things out. So you've basically got an energy gap. And one of the professors in our group did studies giving a type of fat, which is called C8 oil, and I'll explain what it is. 2 tablespoons a day versus placebo to these older people who had memory problems, and they had tremendous improvement in their cognitive energy. They had a 230% increased production of energy in the brain from ketones, because neurons can work on glucose or ketones. So what I'm trying to say here is that it may be too late to stop the total demise, whatever, but there is an energy gap. And giving 2 tablespoons of C8 oil can actually fill that energy gap and restore some mental energy, function, and so on. And it's an inexpensive and very safe thing to do. So fats are chains of carbon. And if it's a very long chain like olive oil, it's 14. And if it's a short chain like ghee, for example, butter, it's like 4. And in between, there's 6 carbons, 8 carbons, 10 carbons, 12 carbons. It's always even numbers. And that's called a medium chain triglyceride. And coconut oil has medium chain triglyceride. And you can even buy MCT oil, medium chain triglyceride. But what we've learned is it actually specifically C8 that the liver makes ketones from. So we've purified from coconut oil, organic coconut oil, just C8, 99.8% pure C8. Coconut oil is only 7% C8. So you'd have to have a lot of coconut to get the same amount of C8. And giving 2 tablespoons, it's best to, the body has to sort of adapt a bit. So start with a teaspoon in the morning, teaspoon in the afternoon, dessert spoon in the morning, dessert spoon in the afternoon, moving up to a tablespoon morning and afternoon, ideally with food. And you may know there are some people who do this with coffee. You know, they call it bullet coffee, where they put it in the coffee. And I've got a thing called a hybrid latte you might want to look up, which is another way of doing this. But basically, for someone who's, got dementia, this can absolutely make a significant difference. So I'm glad you asked that question.
Excellent. I'm going to wrap up now and say very big thank you to you. There's been a lot of enthusiasm in the chat. You'll pick up the chat afterwards. There are some questions which might not have been answered, but we really needed to concentrate on the bigger ones, the ones that had more people asking them. So it's been really interesting. I think everybody's very grateful for you. Quite a lot of people have followed you for a long time, have done your courses in the past, have done other work with you. And I think huge respect to you for everything you're doing. And I must re-emphasize to people, get onto your website, follow up the cognitive test, because that's part of your research. Sign up for the tests and the testing and the ongoing testing, and if possible, sign up for the blood test. And with all of those things, send your results back in. Thank you very much, Patrick. You have an enthusiastic audience who are thoroughly won over. And I think this is going to be a big help with helping people live healthier lives, of which B12 is just one of many contributors.
Well, thank you for inviting me, and please spread the word to everyone you know, especially those over 40. Thank you.
Yes, which is probably everyone here. Thank you.
Bye.
Thank you. Bye-bye.